discussed in our first phase of practical classes, where we talk about the general characteristic, how to study and isolate the bacteria. Like this, we know, like there we mentioned that bacteria we identify on the basis of its cultural characteristics. So almost all bacteria, they had certain unique features and unique characteristics through which we identify them. So yeah, this cultural characteristics, if you can remember what point we talked earlier, is that we study the growth of the bacteria, liquid media, that means in the broth as well as in the solid media. So in the liquid media, we generally look for some specific characteristics if it is present. For example, there is certain bacteria which produces very typical odor. So maybe pleasant or maybe very pungent odor. So depending on that, also we sometimes considered not only the other physical what we can see, even by odor also, like pellicle formations, like flake formations. There are certain bacteria which aggregate when it grow in the liquid media, and what we call as the pellicle formation. The background may be a clear, only certain flakes you can see, whereas the majority of the bacteria, they produces uniform turbidity. So these are also some minor characteristics that we considered while studying the bacteria. This is schematically how we represent, like those aerobic, obligate aerobic, they are in living air. I mean, they are in front of air. That's why they try to grow at the surface. So if we grow the bacteria and without disturbing the culture, if you look, then you can find a obligate aerobic bacteria. They are more concentrated here. This area will be turbid in comparison to the lower part, whereas the obligate anaerobic, they tend to grow away from the surface, because at the surface the oxygen concentration is maximum. Just for our understanding, microaerophile, just simply avoid this aerobic little bit down there, and aerotolerant and facultative organism, these are certain things. Here you can see this is one organism which is forming certain flakes at the top, right? So where uniform turbidity, and here you can see some sediment. So these are also some characteristics, cultural characteristics of organisms. So my point is that cultural characteristic not only in the solid media in the form of colony, but also we study in the broth, in the liquid. So depending on the type of the organisms, where we can record this, and that help us in identification of this organism, right? So that substantially support the information for confirmatory detection of that organism once you grow them in the laboratory media. But remember, it should be of pure culture. Only one kind of bacteria you can study at a time, okay? So most of the time, we consider the growth of the organism in the solid media, where we can generate many information. So what are the informations, like the colony shape, size, elevation? This already we discussed it, but still for your refreshing idea, I'm giving it once again. If you just look the colony from the side, the eye level, then you can find some of the colonies. They are just like a dome. Some are convex, some are almost flat to the surface. Okay, an umbonate shape, and then margin of the colony. Some the colony are very clear cut, smooth, round, just like a complete geometric shape. But whereas there are many where you can see very irregular edges and what we call filamentous and curl. So these are some characteristics of the organisms. If you just try to compare it, like this is one bacteria where you can see a rim is produced, raised rim, and a depression, and the center part is elevated. This is what we call as a raised margin, and this is one umbonate type. So if you know that your bacteria is producing an umbonate type colony, obviously in a mixed populations, we'll try to pick up this particular organisms or this particular colony, or this colony we can correlate with our target organism. So suppose, my point is that suppose you want to isolate, this is, you can see is a flat, a very large, rough type colony. In comparison, these are the smooth colony which are shining, okay? So these are again colonies with wrinkled surfaces. Now, We know usually the Bacillus species, they produces this type of large flat colony, as we talk about the Bacillus anthracis colonies. Now, if you are interested to isolate, or your target organism is Bacillus anthracis, and after inoculation, if you can see a colony like this, then it cannot be Bacillus anthracis. So, likewise, on the basis of the colony characteristics of a target organism which you are focusing, we study how they look like in different media. Okay, not only even like the colony characteristics may vary depending upon the type of media. So we should look for all these characteristics. Okay, like this is a highly fastidious Haemophilus influenzae bacteria, which produces what drop like colony. This is zoom image, very small colony they produce in the chocolate agar media. And the characteristics, the glisteningness, those are criteria that we considered for identification. Now, these are again randomly inoculated some organisms. Well, here also we can see these are some very small dewdrop-like colonies, and all of them they are shining. I mean, glistening. The light is reflecting on the surface of the bacteria. So these are not dull colonies. So if you know, so when we talk about this organism, if you can remember the Streptococcus, we talk about the Bacillus, then Clostridia, all of them, they had their own characteristics. So what type of colony they will produce, like Medusa head colony of Bacillus anthracis, obviously it will be a very big one, not like this. So these are seems to be the Streptococcus colony. So here again, another instance. This is a bacteria which produces swarming type colony. I mean, it's spreading colony. If you inoculate at a point the culture media, just like the sea wave, the growth of the bacteria will be there. And okay, so this swarming colonies. They produce, and finally they cover up the entire culture plate. Right. So now, if you know that the organisms produce this type of colony, obviously it helps us in identification. Some of these characteristics are very, very unique. Here, the example of Yersinia pestis, they produces a sticky colony. Sticky colony in the sense that it adhere and get just like difficult to pick up from the culture plate, and then some of the bacteria where you find their butyrious signature, just like that. If you just try to take out the butter from a butter block, we call butyrious colony, but unlike this is very sticky colony. So these characteristics, like the hemolytic pattern, the blood agar, already we discussed it, right? So the margin, age, everything. Just look at these colonies here. It's totally different. Here the surface is so smooth. Here it is totally rough colony, right? So something like the frosted glass or ground grass like appearance that we can see in certain organisms. So type of hemolysis, non-hemolytic and hemolytic colonies. So these are some of the criteria that we considered for identification of bacteria. Okay. So then it comes again the other parameters, including the opacity. I guess all of you understand about these things. Then pigmentation, yes, like Staphylococcus aureus. Hello.
Sir, someone is waiting.
Someone is waiting. Okay, who is waiting? So this characteristics we study in the solid culture media, different type of culture media, like maybe in the enriched media or maybe some selective media. We record it and we try to correlate with the standard organism. Almost all organisms, they produce a standard type of colonies in a particular media, and then this information help us in identification. But all of you will definitely agree, like this is not sufficient, because colony characteristics of two organisms may appear somewhat similar. So in that case, of course, we cannot come to conclusions about this organism. But in these are the things that we recorded. For your information, I'm giving it. So next, once we can get some idea about this organism, then definitely we have to confirm it by all these different characteristics: coccus, bacillus, arrangement, presence of flagella, motility determinations, then gram staining, whether endospores are there or not. So coccus in chains like streptococcus. So these are like if the colonies are matching the streptococcus, and if you can just demonstrate pictures like this in the gram staining, then definitely we can say streptococcus. But question arises whether which species of streptococcus. So Pyogenes, Agalexi, Disagalexi, Equisimilis, etc., Zooepidemicus, you can say. So for that reasons, we have to depend on the biochemical test. So on the basis of the biochemical test, of course, we can come to a conclusion and identify them. Okay, so that will be the main focus for today's class. Although some characteristics like this endospore bearing Clostridia in the muscle impression smear is a clear indications of black quarter. So some cases like this is the drumstick bacilli of Clostridium tetani, no doubt it is quite easier for identification, but more or less most of the organisms we have to go for biochemical tests for confirmatory identification, right? So this is again the bipolar Pastorella organisms in between the blood cells in the blood smear. That I hope this slides already if you have seen it earlier. Just for refreshing your idea, I'm just showing it. Then come to the biochemical test for identification of bacteria. Okay. So here what I'm mentioning repeatedly is that among the two closely placed organisms, that means two species within the same genus, they differ from each other. Their name has been given specific name only because there is some difference among these organisms, and these differences we can explore on the basis of the biochemical characteristics. Sometimes even on the basis of antigenic characteristics also these demarcations were prepared. Okay. Well, sorry, a lot of disturbances coming from my side. Okay. So this biochemical test we perform for identification of bacteria, as I was telling it, on the basis of some antigenic characteristics, also some strains within the same species or serotypes are identified. But in general, for confirmatory identification of bacteria, we perform the biochemical test. Remember, this for bacteria and few fungi, this kind of tests are available, but not for viruses and other, because the concept of viruses is different. Right? So, as such, there are many biochemical tests developed for specific organisms for their identification purpose. And yeah, this the list is increasing as newer and newer organisms are exploring, and the best way, the easiest possible way to identify those characteristics, differences in the biochemical behavior of the organisms is day by day is incorporating into the list. So as such, what we feel is that it's difficult to understand each and every biochemical test and their principle, but to some extent, some of the biochemical tests which are most commonly we use it, and even we discuss it for species level identification of the organism of our interest. So a sort of classification, means grouping, has been made, like those biochemical tests which are based on breakdown of carbohydrates are grouped in one, so that we understand that their main principle lies with the breakdown of sugar, and that within that category we have three different type of tests: sugar fermentation, methyl rate, bogus Prescott. Of course, many more may be there, so we'll be restricting to these three tests. Then some other tests are again based on breakdown of proteins. It has been seen that the bacteria differ from each other on the basis of the breakdown of proteins in the metabolic pathways. Some special molecules are formed, some enzymatic activities are performed, and because of which some changes in specific media we could observe, and we can declare it as a positive or negative. Likewise, where we can go for a gelatin liquefaction, indole production, ammonia production, then nitrate reduction tests, etc., we can perform. Okay. Then, of course, there is a category called the miscellaneous tests, where many different type of tests are grouped, and mostly the enzyme activity of the bacteria are assessed in those tests. Okay. I'll just take one more minute. Is it here now? Well, we'll refer to this particular slide. So as such, we'll try to discuss this few of those biochemical tests for identification of bacteria. Can you see the slide properly?
Yes, sir.
Okay. So the first category is the biochemical test on the basis of breakdown of carbohydrate. So here the first test is a sugar fermentation test. This is a very commonly used test, and since for a long time people describe this particular test for differentiating important members within the same genus, the species level. So the bacteria, they differ on the basis of sugar fermentation. So principle says that the bacteria, they're able to ferment the sugar, and when they ferment the sugar, there is production of acid. So when acid production will be there in that appropriate sugar solution, then the acid formation can be detected by a certain pH indicator. You put few drops of the pH indicator, and color change will take place, and you will come to know whether acid formation is there or not. So it's a very simple test. Simply, we need to prepare the sugar solution in some peptone water or in the nutrient broth, and then we inoculate the pure culture of bacteria and incubate overnight. So after overnight incubations, we test whether acid formation is there or not by adding certain pH indicator, like Android reagents is very commonly used in this case, and this gives us intense red color in the acidic pH. Okay, so at the same time, so it has been seen that certain bacteria, when they ferment sugar, they produces gas also. Whereas at the same time, our close organisms to that may not be producing gas. So gas formation by sugar fermentation, then only sugar fermentation, no gas formation. These are the certain characteristics that we can find it out by sugar fermentation test. Okay, so for that we need a special device called as the Durhams tube. I don't know whether it will come or not. Yeah, I prepared this particular video in our lab to show you how to insert this tube, and we can understand how it works. So suppose this is a tube, and these are the Durhams tube. Okay, please see it very properly. It's look like the test tube, just like the miniature form of the test tube, very small tube, glass tube. These are called as the Durhams tubes. Okay, and these Durhams tubes are inserted into this media invertedly. Okay, so it look like a test tube only, but it's a very small one, right? So we prepare some sugar solutions, and then we put the sugar solutions into the test tube, right? So at the same time, so we pour your sugar solution, the same media, to the Durhams tubes also. We have to fill it up very properly. Okay, so there should not be any formation of gas bubble at the top. very carefully we need to fill it up. Just see here, this Durham's tubes are filled up with the same media. Now we need to insert it invertedly. So you see the things here like invertedly. Okay, so this is a tube, invertedly we have to insert it, right? After that, this is the situation here. So this way we insert the Durham's tubes into this media. Then we inoculate, then we definitely we have to autoclave the whole things, and then we have to sterilize it, and after that we inoculate the bacteria. So when we inoculate the bacteria, suppose this is the situation here. So bacteria will grow here, and bacteria will also go inside these Durham's tubes. Whatever media is there, there also the bacteria will grow. And after overnight culture, we put the enriched reagent to see whether there is any acid formation is there or not. Here it is showing this tube, there is acid formation. Here the bacteria is growing here, but there is no acid formation. So because enriched reagents doesn't produce any color in this particular tube. So here there is acid productions and there is no gas formation. But in this case, this particular bacteria, they able to ferment the sugar and produce the gas. The gas that produced inside the Durham's tubes cannot escape because you have inserted invertedly. So they get accumulated, and this indicates that this particular bacteria can ferment the sugar as well as can produce gas. Maybe it is a glucose, fructose, dulcitol, mannitol, whatever. 100 different types of sugars are available, and we need to select it like our focused organism or target organisms, which are the sugar help us in close differentiation with other related species. So accordingly, we need to prepare a panel of sugar tubes, and we have to go for performing the sugar fermentation test. So usually we grow the bacteria in a broth culture, overnight broth culture, and then we instill few drops of the overnight culture, and then further we incubate, and next day we perform the test using the enriched reagent. Okay, so it's this is another tube is showing a gas formation inside this Durham's tubes. So likewise, we can perform it. Now this sugar fermentation test, as I said that the example I have given it for single sugar. So we have to prepare the sugar solution individually. So you can select 5, 6, 10, 15 different sugars. Now how to select this sugar? It's all depend on which organism you want to identify. Suppose you have suspected that might be the Streptococcus agalexi. So you selected what are the sugar that used for differentiating Streptococcus pyogenes, agalexi, and other species. And then you select that particular panel and perform this test and record it positive, negative, means sugar fermentation positive, negative. And finally, you match with the standard search to find it out whether it is matching with Streptococcus agalexi or not. So this is the way how we interpret it. So the second test within the same category, breakdown of carbohydrate, is termed as methyl rate test, or shortly we call it the MR test, methyl rate test. Why your understanding of this test is important? Because in the last few couple of classes, like when we talk about the Staphylococcus, we talk about Streptococcus, we mentioned about the catalase test. So you will be learning today how catalase test gives us how it is performed and what type of interpretation we can get it. So similarly, in the subsequent classes, you will be getting the name of methyl rate test. So MR test is another important test, usually for identification of enteric pathogens. We perform it, but this is not only for enteric pathogen. For some other organism also you can perform it. So methyl rate test is based on fermentation of glucose. It has been seen that there are certain bacteria which can ferment glucose and can produce heavy amount of acid. Heavy amount of acid means the pH goes up to 4.5. Usually, many bacteria which cannot produces acid up to 4.5 pH. Maybe it's remain as 5.5 or up to say something less than that. but not up to 4.5. So those organisms which are able to ferment sugar glucose and produce the pH up to 4.5 and below can be detected by this particular test. Here we use a pH indicator which is called as the methyl rate reagent. This methyl rate reagent, it produces red color at the pH below 4.5, and above it produces yellow color. Yeah. Please microphone. Please control your microphone. Rida Fatma, Anime Sil. Please control your microphone. Well, so we prepared sugar solution, glucose solution, not sugar. It's, I should specifically say glucose, because this is based on fermentation of glucose. So buffer call as the glucose phosphate peptone water is prepared, and then we inoculate the bacteria and we grow them and incubate them for 24 hours. And after that, we add few drops of methyl rate reagent. If the color remain red color, then indicate this is positive for heavy production of acids, and if it is yellow, then it is negative. So likewise, we can interpret whether organism is positive or negative by methyl rate test. Then at the same time, another test, shortly we call it a VP test, Voges-Prescott test. Voges-Prescott test. This particular test, again, it is somewhat related to the methyl rate test. It has been seen that among this important member of Enterobacteriaceae, they can ferment glucose, can produce heavy amount of acids, but those acids are further metabolized by a group of bacteria to neutral compounds. So initially they can ferment glucose and produce acid, and that the acids are further converted to a neutral compounds like acetyl-methylcarbinol or 2,3-butylene glycol, etc. So those kind of bacteria can be identified by Voges-Prescott test. So we use the same GPPW glucose phosphate peptone water. We incubate the bacteria, but in this case, we give a little bit prolonged incubation. So may go up to 48 hours of incubation. After that, we use alpha naphthanol solutions along with potassium hydroxide. So these potassium hydroxides will lyse the bacteria, and these neutral compounds will come out. And then when they come in contact with 5% alpha naphthol, then it will give rise to color development. So if there is formation of red color here, then it's indicated it's a positive VP reaction, and if it is yellow, then it is negative. So likewise, simple overnight test or within a day or two, we can perform it and get the results. So Voges-Prescott test, shortly we call it the VP test. The principle lies with the bacteria able to ferment glucose, produce acid, and later on the acid is neutralized to certain neutral compound, converted into neutral compound. So those are called as the Voges-Prescott test. So this is again based on the breakdown of carbohydrates. So these are the three tests: sugar fermentation test. Sugar means different type of sugar. Then methyl rate test and Voges-Prescott test are included into this group. Fine. Then coming to the next category, it is the reaction due to breakdown of proteins. So in this we get several type of tests like gelatin liquefaction, then indole production, ammonia production test, nitrate reduction test, urea hydrolysis test, and hydrogen sulfide production test. A few of it I just selected from the list and incorporated here. So we'll discuss it because this principle is lies with breakdown of protein. As you know, like first test is a gelatin liquefaction test. If you can remember, when we talk about the anthrax chapter, we discuss about this inverted Furi-3 appearance. It's because in the gelatin media, The Bacillus anthracis is a slow liquefier of gelatin. So the step culture look like an inverted fig tree, whereas the anthracoids, anthrax-like bacteria, environmental contaminant, which are a prolific gelatin liquefier. So they can liquefy the media totally. So if you can remember, when we talk about the historical development of microbiology, there we mentioned about that initially when the Robert Koch and Louis Pasteur was trying to isolate the bacteria in the pure culture, then they use this bovine gelatin as a solidifying agent. Because this bovine gelatin, when it dissolves in water and then boil it and cool it down, then it will give you a solid masses, okay, just like a gel-like consistency. But the biggest difficulties they face is that those bacteria which are gelatin liquefier, they have the gelatinase enzyme and can liquefy gelatin, bovine gelatin. Once the colony development takes place, the whole solid media become liquid and they cannot go for the pure culture. So that was the greatest hindrance they face it. And finally, the Fanny-Hasse story, you understand how they incorporated agar as a solidifying agent as a bacterial media. Until now, we are using agar as a solidifying media. So in this gelatin liquefaction test, we prepare the bovine gelatin solutions and then we sterilize it and just prepare just like in upright positions. We allow it to solidify in the tube. Okay, then not in the slope. Then we inoculate the bacteria, and if the bacteria able to liquefy the gelatin, you can see the pert will become liquid. So it can be best seen if you just tilt the tube like this way. So the gelatin liquefier or non-liquefier can be find it out, and it's based on breakdown of protein. Then the next test is the indole production test. This is another very frequently performed test, and mostly for Enterobacteriaceae identification of important members like Escherichia coli, Salmonella. Then even like some Proteus and Pastorella, they also show positive, where the E. coli is positive and most other enteric bacteria, they are negative for indole production. So this test is based on breakdown of amino acid tryptophan. Please note it down. This is a very frequently asked question. It has been seen that there are certain bacteria which can break amino acid tryptophan. Remember, only the tryptophan, and then that decomposition of tryptophan leads to production of a compound called as indole. So indole production inside the tube or the media can be detected with a color reaction, and for that we use a substrate. It's called as the para-dimethyl aminobenzaldehyde. Shortly, it's called as Covex reagent. So this Covex reagent, it is straw in color as such, but once it come in contact with the indole, then immediately it will convert into deep purple color or deep pink color, you can say, and they float at the surface of the bacterial culture, and we can say that this tube contains this indole. This is the picture, and for that we prepare peptone water which sufficiently contain the tryptophan amino acid, and then we inoculate so overnight, and the next day we put few drops of Covex reagent. So this Covex reagent, once it come in contact with the indole, then they will convert into intense pink color ring at the surface of this media because it contains some isoamyl alcohol. So they simply float it. Whereas the tube where there is no production of indole, it remains straw in color. It's just like the brown color they will develop. So it's a very simple test can be performed for detection of indole production. So, you know, like there are many organisms which specifically produces indole. Among the Enterobacteriaceae, it is the Escherichia coli that produces indole, and that become a hallmark in identification of Escherichia coli. Always we perform this indole test to differentiate with other member. Okay, so the test is principle is based on breakdown of protein. Similarly, the ammonia production test. So it has been seen that few bacteria, they produce enzyme deaminase, and they can break amino acids and can liberate ammonia. So they act on the amino acids and they can break these amino acids and they can produce ammonia inside the tube. So as you know, this ammonia is a highly alkaline substance. So very easily we can detect the ammonia production so with some pH indicator. Okay. So what basically we do, we use some phenol red indicator, and the color will turn yellow to orange, and in negative reaction there will be no color development. Okay. The next one is the nitrate reduction test. So this test is also based on breakdown of proteins. So here the basic principle says that these organisms containing the nitrate reductase enzyme, which can cause conversion of nitrate to nitrite, and finally nitrogen dioxide. So this conversion is possible with the nitrate reductase enzyme. So only those bacteria which possess this enzyme can be detected by nitrate reduction test. Okay, and this nitrate formation can be detected with the color reaction. So again, red color development takes place with a specific indicator, and the indicator we use an alpha-naphthylamine, okay, and sulfonylic acid combination, and there is formation of red color of nitrous NO3, and if there is conversions, then it becomes colorless. Okay, so nitrate reduction test from the left is uninoculated, the positive for nitrite and negative for nitrite. Okay, so here there is no conversion, and here conversion takes place. These are some easily performed tests in the laboratory conditions, and routinely people use this kind of test for identification. The next is the urea hydrolysis or urease test. So there are some very important organisms, and for their identification always we perform this particular test. Like we prepare the media containing urea, and then presence of urease will convert this urea to ammonia, and the ammonia will change the pH indicate pH color of this test, and like here it is showing that this indicate this alkaline reactions are produced. The phenol red indicators are here, and the yellow color indicator is no negative for urease production. So we use a particular media. It's called Christensen's urea agar media, which contains a sufficient amount of urea, and then we inoculate the organisms, and presence of this indicator will give the color formation if there is formation of ammonia. Ammonia is highly alkaline, then this is an alkaline reaction showing it here. Okay. So again, at this point, I should tell you, like when you prepare this Christensen's urea agar media, means urea is added to the agar media, and you cannot heat up the urea. If you heat it up, then it will destroy. So just like the blood agar, how we prepared it, the urea solutions are also available in the sterile form or incorporated to the basal media at a temperature of 50 to 55 degrees centigrade. The next is the hydrogen sulfide production. I guess all of you during your 11 and 12 standard, you perform the test for detection of hydrogen sulfide, and there you learn it that the ammonium sulfate, which is a white salt, when come in contact with hydrogen sulfide, they will convert it into black color. At the same time, the other is the lead acetate, ammonium sulfate as well as the lead acetate. So here in this test of H2S production test, we prepared lead acetate paper. Like we prepare saturated solution of lead acetate, then some paper strips, filter paper strips are soaked in lead acetate, and we dry it and keep in the laboratory. So when we want to taste it, then what we do, we inoculate the bacteria into a vessel media, mainly the peptone agar media, and then we attach a lead acetate paper at the top of this tube and incubate it. So if the bacteria is able to produce hydrogen sulfide by degrading the sulfur containing amino acids, then this lead acetate paper will become black. That indicates it is positive H2S. So these are some of the tests which are based on the reaction due to breakdown of proteins. Okay, I guess some idea you're getting how they work, and you have to go through all these different tests and their principle. Then coming to the miscellaneous category, where certain compound tests like catalase test. So we mentioned about this particular test to differentiate Streptococcus and Staphylococcus. As you know, like Strepto, they are negative, and the Staphylococcus are positive for catalase enzyme. So this particular test is for detection of presence of catalase enzyme in the cell wall of the bacteria. So it's a very simple test. Simply, we need to put a few drops of hydrogen peroxide at a glass slide, and we pick up our bacterial colony and mix it up with the hydrogen peroxide. You know, this catalase enzyme will immediately break hydrogen peroxide, and this hydrogen peroxide will convert to water and oxygen. And this oxygen form, because of this breakdown of hydrogen peroxide, comes out from the water phase very rapidly, and you can see just like a formation of froth like this way. And this is what we describe as the effervescence formation. So it's a very simple test. You take a glass slice and take hydrogen peroxide, a few microliter, and then you pick up a colony and mix it up. Immediately, if there is effervescence formation, that indicates this bacteria is positive for catalase enzyme. Whereas in this side showing the bacteria after mixing, also there is no any effervescence formation. That means this organism is negative. So among these Strepto and Staphylo, the Strepto are negative and Staphylo are positive. So if you had any confusions in the staining and cultural characteristics of these two bacteria, no doubt we know the Strepto are the coccus in long chain, but sometimes what happens when you prepare a bacterial smear and stain it, during this process, physical aggregations or disintegration of this bacteria, sometimes they may auto-aggregate and may be confused whether Strepto or Staphylo. So in that case, if you had a pure colonies, we can go for this test and you can confirm it. Okay. So same test even we can perform in the culture plate, where we can pour a few drops of hydrogen peroxide over the colony and you can just see how this effervescence are producing. So this is the catalase test. The next is the oxidase test. This is another test very frequently performed, and we mentioned in our previous lectures about this organism is positive and negative for such and such enzyme. So this oxidase test is another very frequently performed test for inhibition of bacteria. Here the principle lies with the presence of oxidase enzyme on its surface. So these oxidase enzymes are allowed to react with our substrate. This is called the tetramethyl paraphenylene diamine. This is a colorless compound. So this colorless compound, when they come in contact with the oxidase, immediately deep purple color formation takes place. So we can get the result instantly. So to perform this test, what basically we do, we use certain paper, filter paper soak in tetramethyl paraphenylene diamine. And then these paper strips are used for identification of oxidase positive bacteria. So you pick up a pure colony and then we streak it in that paper strip. So after streaking, we need to see the development of deep purple color. And if this color development takes place, remember, within 20 seconds, we need to record the test result. So if it is delayed, then there may be a chance of false positive reaction. So this conversion takes place instantly. Within 20 seconds, we need to observe the formation of deep purple color. So same test even we can perform in the culture plate, where we can prepare the solutions and then we can pour it. Those organisms or colonies are positive for oxidase enzymes. They will produce deep purple color in the media plate. So these are the most commonly performed biochemical tests for identification of bacteria. This is another like methylene blue reduction test, and basis is for detection of dehydrogenase enzyme present in the bacteria. And this is an color changing this bacteria can change the color of methylene blue and leucomethylene blue, which is a colorless compound. So the methylene blue, you just inoculate these organisms, and after overnight culture, you can see the color will get reduced. No color formation takes place because of this dehydrogenase enzyme. So these are a simple test, and bacteria, they differ from each other on the basis of this test, and they are helpful in identification purpose. This is another test called as the citrate utilization test, and this is also really important in identification of important members in the Enterobacteriaceae. Here again, the principle says that there are certain bacteria which can utilize inorganic salt as a sole source of carbon and energy. Like what we learned in our earlier classes is that the majority of the bacteria, they are heterotrophic in nature, in the sense that they need the reduced organic compounds like carbohydrate, then protein, then fats. They need it for source of energy and carbon, and they can grow and multiply. But we call them as the heterotrophs. But there are certain pathogenic organisms which can grow and multiply in presence of inorganic salt. They don't need any organic compound, no reduced organic compound. They don't require even the inorganic salts like sodium citrate. They can grow and multiply. Some ammonium citrate also they can utilize as a sole source of carbon and energy. So to identify those bacteria, among these important pathogens, I can remember this Salmonella is a citrate positive organism. So this typhoid causing bacteria, always we perform this citrate utilization test, and we prepare a media called as a Simon citrate containing the pH indicator. And this pH indicator in the normal pH of range 7 to 7.4 is green in color, but when it becomes alkaline, then it will become blue, deep blue color. So this particular test, we prepare a Simon citrate agar. It doesn't have any organic compound or nutrient. Rather, it is composed of salts, ammonium salts, and then we inoculate the bacteria. And most of the bacteria, they cannot grow here. Cannot grow here because it doesn't have any organic compound. But those bacteria like Salmonella, they can grow and multiply in presence citrate, and then the pH become deep blue color. So this is the citrate utilization test. So likewise it progresses, and this is I guess the last one that I'm going to talk about. In the pace of development, what happened, people try to explore incorporation or single tube biochemical test to generate more information. So as such, a particular media has been developed. It's called as the TSI agar media, triple sugar iron agar media. So 3 sugars like glucose, lactose, and sucrose are present in this particular media at different concentrations. Then a lot many other compounds like phenol that is the indicator. Then it has then some ferric ammonium citrate and sodium thiosulfate for detection of hydrogen sulfide productions inside this tube. Okay, so what happened in the uninucleated tube, which is usually remain alkaline state, it's become red in color, TS agar media. This is uninoculated tube. Okay, so certain organisms, they can grow here, and if the slope is red color slope, this is the slope, and this media is prepared in the form of a slant. If the slope is red color, this indicates that this bacteria cannot ferment lactose, but they can ferment sucrose and glucose. And the butt is yellow. And formation of black color indicate this organism is also positive for H2S production. Here you can see same media. This is the uninoculated tube, and you can see H2S positive, lactose negative. Here, negative, and glucose and H2S, and all the three sugars you can formation. And this really simplified the process of identification. And interestingly, you can see it here, even like certain bacteria which can form gas during the fermentation of sugar. Can be seen it here. This medium may be pushed up or maybe get cracked. That indicate the bacteria have the ability to produce gas also. So these are the different biochemical tests that we use it for identification of bacteria. I guess all of you have this book with you. If not, this book can be downloaded from internet, and please go through it. It is having specific bacteria, all the biochemical reactions that are recommended for identification purpose. So in bacterial isolation and identification process, always we take help of this type of standard book with good pictorial demonstrations, and we compare, because it's not possible for us to remember each and every biochemical test exhibited by all these different pathogenic organisms. So we take help of the standard references for identification purpose. So this is about this biochemical test for identification of bacteria. But this process is, suppose you want to perform all these different biochemical tests, it's really a laborious process, time consuming, and a lot of culture work you need to perform it. So these things has been converted into certain automatic form with the development of certain high cost equipment. It's called as the automatic bacterial identification machine. So one such machine is available with us in our department. It is the BD Phonics equipment, and within 10 hours you need a pure colony of gram-positive or gram-negative. Up to that part you need to identify. Then you put it into the machine in certain special cassette. Within 10 hours it will perform several different, almost 50 different biochemical tests it perform, and then it will identify by reading the algorithm of all the results, and it can identify the organism into genus and species level. Of course, in the database all the information should be present, then only you can identify it. So this machine comes along with the preloaded software which contain all the informations of whatever known organisms present, along with your biochemical test results. And once the results are detected by the specific sensor present in the equipment, then they compare with that and then they identify the organism. So simply what we need to do is to isolate the bacteria in pure form, and simply we need to test gram-positive or gram-negative. And accordingly, we had a panel for gram-positive, we had a panel or cassette for gram-negative. So we can simply inoculate a few organisms and then it will automatically identify it. So this is a fantastic system available with us. Once you'll be returning back, then we'll give you a glimpse about this equipment, how it works. And through that, we identify so many unknown bacteria from environment as well as from gut of different species. But all this system, it's not like something different. It's only the biochemical tests on the basis of different biochemical tests. Almost 50 different biochemical tests are performed, and they will be belonging to one of the other group as we discuss it today. So we selected few of the important biochemical tests for your understanding about the bacterial identification system. So at least I believe that all of you at the end of this class will get a comprehensive idea about how a bacteria from the environment can be isolated in pure form and can be identified. And that is the main intention. You must keep in your mind the whole process of isolation and identification. Of course, this is that traditionally we are learning all these things, but as this generations, a lot of advancement in the genomics has taken place, where gradually the cost of whole genome sequencing become easier for the people to do it. So if we had a bacterial isolate and give the entire genome sequence, and then using certain online software, we can very accurately identify the bacteria, which has revolutionized the whole process, and many laboratories they are now adopting that kind of practice. We hope that these things will be of easy access to all of us very soon. So recently from state of Mizoram, in the Mizoram University, they have sequenced one organisms in their own lab. The whole genome was sequenced, and probably in near future, this technology will be well accessed to all of us, and that will, of course, revolutionize the entire process of bacterial identifications and characterization. But at the same time, we must understand the principle of this test also, because at present, on the basis of this test system, we identify the bacteria. So this will be the beginning of the systematic bacteriology. Always before starting the discussions on individual bacteria and their disease and how we diagnose, we must go through this biochemical test, because suppose for identification of bacteria, if you mention that we perform this, the organism is oxidase positive, then you should be in a position to understand what do we mean by oxidase positive. So all informations are available in this particular lecture. Okay. Thank you very much. Do you have any query or any questions? Of course, in the next class, definitely I should come up with this lateral flow test. Somehow it got out of my mind and slides are not available. So these pictorial diagrams are important to understand the lateral flow assay. At any point, we can discuss it in the subsequent classes. Yes, any question, anything, or you can just text me. So if no query, no questions, then we'll meet in the next week. Thank you very much. And closing the link. Thank you.
Thank you, sir.
Thank you, sir.
Thank you, sir.
Thank you, sir.
Thank you, sir.